Okay, good evening everyone and welcome to Genomics Lite. This is the third in our series of human genetic variation. So it's our careers pathways panel, and we're really lucky to be joined by 4 different researchers that all work on something related to human genetic variation that are going to be telling you a little bit about their career and their work at the Wellcome Genome campus. Before we start, just a couple of things to mention in case this is your first time joining us on one of these webinars. We're obviously using Zoom webinar, which means that you can't use your camera or your mic today. So we can't see you, but you can hopefully see us. If you need to get hold of us, if you're having any tech issues or you have any questions, there's a button at the bottom that says chat, and you can use that to say hello to us, to ask any questions you might have. And if you're having any tech issues, you can let us know there. If you've got any science questions or questions about careers for our speakers, you can use the Q&A button at the Q&A button at the bottom as well, and that just makes it a little bit easier for us to find your sort of Q&A questions later on. We're also using closed captions this evening via a service called Otter AI. I know that these aren't perfect. We've only just started using them. If they're in the way and you don't want to see them, you should be able to hide them using just the little arrow button. So if you want them, they're there. If you don't want them, you can get rid of them. So I think that's my little blurb about the webinar. I'm going to share my screen again and tell you a little bit more about the campus in case you've not heard of the Wellcome Genome campus before. So as I mentioned, this is the third in this series all about human genetic variation. Next week will be our last in this series, and we'll be looking at the big questions in human genetic variation with Richard Milne, and there'll be links for how to get involved in this in a second. So we all work at the Wellcome Genome campus, which is based just outside Cambridge in England. And so I'm interested to know where all of our audience are from today. I've seen someone put that they're from, I think, Canada. But if anyone else wants to tell us where they're from, you can use a chat box to let us know. I think that was California, not Canada. Cambridge, London, oh, Cyprus. I hope it's a bit nicer weather in Cyprus. Maryland in the States. Kent, Norwich. Okay, we've got quite a mix this evening. So welcome to all of you. I think California is looking like the furthest from campus today. So as our name suggests, all the groups on campus look at something to do with genomics. We have 2 main research groups on campus, the Wellcome Sanger Institute and EMBL EBI. And our researchers that are talking to us this evening we've got a mix from both institutes, which is really exciting. So I'm going to put a quick poll up on the screen that's going to ask you how much you know about genomics. And that's not like a test or anything. It's just because we obviously can't see you. So it's our best way of knowing who's in the room this evening. So this should pop up on your screen and you can answer that on the little poll, or you can put a little note in the chat box if you'd rather. Awesome. So we've got a bit of a mix, but quite a lot of you have a fair or good knowledge of genomics, which is really nice to see. And hopefully by the end of this session, you might know a little bit more, especially about sort of career options in genomics. So whilst everything we do on campus is to do with genomics, we have 6 main research areas, which are human genetics, cancer and mutation, cellular genetics, parasites and microbes, the tree of life, which is a project looking at biodiversity in the UK, and biodata. So we've got a sort of mix of stuff this evening, but mainly fitting in the human genetics area and the biodata area. I think that will be all from me for now. I'll pop some links in the chat in a second to some resources. If you're studying for any school or university exams, your Genome's a really great place to find activities, videos, resources about lots of things to do with genomics. And if you want to know when our future series of Genomics Lite become available, you can sign up for our education update, which I'll put a link to as well. So I will stop sharing my screen. And I will hand over to our first speaker, who I believe is Hilary Martin. Hilary, do you want to introduce yourself? I think you're on mute just there.
Sorry, just trying to find it. Okay. Can you now see the main screen?
Yeah, that looks great.
Good. Okay. Thank you, Em. And I'm excited to be here. I haven't done one of these talks before, so I hope it's semi-interesting and useful. I run a lab at the Wellcome Institute in the Human Genetics Department, and I'm going to tell you a bit about my career trajectory. So I was born in Australia, in Brisbane, this city in the middle of the East Coast, and I went to school and did my undergraduate there. Between school and undergraduate, I spent a year in France where I tried to learn languages and history and generally enrich my knowledge of the world before going on to study science at university. And I did that at my local university in Brisbane, which is pretty common for Australian undergraduates. Although in the middle, I did 6 months at the University of California, Berkeley, which was a really formative experience. And at the end, I did a year, which we call honours in Australia, which is a pure research year. So it turned into about 4.5 years of undergrad. And then I came to the UK in 2011 to do my PhD in Oxford. After that, I came to the Wellcome Genome campus and did a 2.5 years of postdoctoral study, postdoctoral research at the Sanger Institute. And then I started my own group within the Institute about a bit over 2 years ago. So I realized this will be different from the schooling most of you in the UK will have had. But in Australia, it's common to do 6 subjects in the last couple of years of school, and these are the ones that I did. So you'll notice there was no biology, and as you'll see in a minute, I did nonetheless have some biological knowledge when I went into the subject of genetics. I went to the University of Queensland, which is my local university, and I had a pretty general first year of maths, chemistry, physics, and a couple of different areas of biology. And the genetics really turned me on quite early, which was probably predictable because I was very lucky that both of my parents are actually geneticists. And so I had had a lot of exposure growing up to discussions about exciting research that was going on in the field. And I was already pretty interested in the subject. But what really got me excited was a figure like this, actually, which those of you who have done some genetics in school might recognize. So basically, this is just indicating how if you have a single gene controlling a trait, you might have this, or you will have this kind of discrete distribution of phenotypes depending on the genotype of that single gene. But as you add more genes contributing to the trait, you converge towards this more continuous distribution of phenotypes. So most phenotypes we care about, like height or BMI and cognitive ability, follow this kind of normal distribution, and it's a consequence of there being many, many genes, each of small effect contributing, and that can be described with statistical model. And I was really excited when I started to see how biology could be combined with maths in this area of genetics, and combined with the genetic and environmental influences from my parents, I decided this was the field I wanted to pursue. So I did more genetics in my second year of undergrad, as well as studying probability and statistics, which are a really important component. Then in my third year, which I spent partly in California, I did more genetics and some kind of harder core statistics. And that was an extremely valuable experience because I did some graduate courses that were intended for PhD students, and I really challenged myself and learned a huge amount. So it was definitely worth going somewhere different with really high standards for that part of my education. And then I came back to Brisbane for a year to do a year of research for what was called the honours year. I actually did quite a lot of research during my undergrad, starting off with pretty menial work, as all of us do, and quite molecular work in a lab. I spent a summer at a lab called Cold Spring Harbor in New York State, and that was really formative because I interacted with people from many labs and learned about multiple areas of biology that I knew nothing about from my undergraduate classes. So it was a really exciting time. it was also very important because I realized that actually I hated lab work and I wasn't very good at it. So if I wanted to do genetics as a career, then I needed to learn to do computer programming, because the field was generating huge volumes of data and you can't analyze those in Excel. You have to learn to program. So in my third and fourth year, I made sure to do research projects that would allow me to learn to program. And I did that quite a lot of it on patterns of gene expression from various angles, and some work in Drosophila. So by the time I came to apply for my PhD, I'd been exposed to quite a few different areas of research within genetics, and I decided to go to Oxford and to study at this place called the Wellcome Centre for Human Genetics. And I worked on a pretty broad range of projects, ranging from rare disease through meiotic recombination to, believe it or not, population genetics of the platypus. So clearly nothing to do with humans, but we were studying, we were doing whole genome sequencing of this very cute creature to learn about the population structure and evolutionary history. After that, I came to the Sanger Institute for the first time in 2016 to do postdoctoral research. And then I really kind of homed in on rare disease genetics, working on this fantastic project called Deciphering Developmental Disorders. And in fact, Matt Hulse, who runs that project, gave a sort of public webinar yesterday where he would have talked about this. And I think that's available on the Sanger website if you're interested to hear more. Then I started my lab at the Institute in September 2018, and this is us, still quite small, 6 people, but it's an exciting time, and we're working on quite a range of projects, still on developmental disorders, but also kind of branching out from that to look at the genetics of cognitive and behavioral traits more generally, because it turns out studying cognitive ability and mental health traits in the general population is important for understanding the causes of very rare, severe neurodevelopmental disorders. Then quite a lot of our work is focused on the genetics of South Asian populations, looking at the basis of complex or polygenic traits, as well as the population structure and evolutionary history and how that influences disease risk. There are pros and cons of every job, of course, and some of the things I like best about mine are when my student or postdoc comes in with some exciting new results that they want to show me. They've got a lot of new plots, it's always a good sign. I also like managing to help them solve technical problems, of which there are many, and they can soak up a lot of time, so it's very satisfying when you finally solve them. And a third thing I really enjoy about it is attending conferences to hear about exciting new research from all around the world. And it's a very international field. And so you kind of make friends from all over the place, and you get to meet up with them once or twice a year at the usual conferences. And it's a lot of fun. One aspect of the job that is less fun is the kind of bureaucratic aspects. Setting up studies requires quite a lot of paperwork, including increasingly legal agreements, which can be quite time consuming and sort of feel like they're holding back the science. But, you know, on the whole, I really enjoy my job, and don't let things like that put you off. It hasn't been completely smooth sailing, although clearly I had a major advantage in that both my parents were working in the field. Just to give you some examples of setbacks, at the end of high school, I really, really wanted to come to Cambridge to study natural sciences, and I aimed high and I applied for Trinity College, which is possibly the most competitive college, and I was rejected. Which was very disappointing at the time, and that's really why I ended up going to my home university, which was not a very adventurous thing to do. But it turned out actually 11 years later, I returned to Cambridge, and I was funded during my postdoc by a research fellowship at St John's College, which is right next to Trinity College, and I got to live for a couple of years in this pretty amazing building, right in that tower there, looking down on Trinity College next door. So I came a full circle. This was another temporary setback. When I was applying for a PhD, I started to write to labs around the world that I was interested in. And I wrote to this lab head and said, I really like what you're doing, and could I come and meet you and talk about your research, because I'm going to be in that part of the world. And I sent him my CV, which I thought was pretty decent. And I received this fairly crushing reply: sorry, too busy, don't have time to meet you. Well, 8.5 years later, this person hired me into my first faculty job. So don't be put off by people like giving you these disappointing responses. Just, you know, work hard, and if you think you're quite good at what you do, don't give up, and you'll be rewarded in the end. So what advice would I give to those of you interested in entering this field? Probably the most important is do more maths and probability and statistics when you get to university. I really regret not having done more. I think you can do much more interesting stuff in this area if you're well trained in this. Definitely learn to program, and ideally do it while you're tackling a research question you're interested in, rather than just using some kind of abstract textbook, which I found quite boring. Make sure you get undergraduate research experience, and when you come to apply for a PhD, you should have a reference from at least one research supervisor, not just from your undergraduate tutors. And finally, prepare for setbacks along the way. There's lots of failures in academic careers. You'll have papers rejected all the time and grants rejected, and you have to be resilient to those. And also you'll have research, you'll have analyses you're trying to do, or experiments that fail most of the time, and you have to be quite persistent. So with that, I'll finish and take any questions.
Awesome, thanks so much for that, Hilary. There was one question about you spoke about using like computational methods in your genetics, and so someone's asked whether there's still a way of doing genetics without doing the sort of coding and data analysis side.
So there is, and I think you're going to hear from some of the other people on the panel about the very interesting and important work that they do, which is actually generating the data that I analyze. So yes, there's definitely a place, you know, this a very important role for wet lab work still, but I think that the computational part of the field has expanded hugely in recent years. And if you want to be able to, you know, analyze very big data sets, you are going to need to do coding yourselves.
Awesome. That was the only sort of specific question for you. So I think we'll move on to our next speaker, but if there's any more general questions, we'll have time at the end that Hilary and the rest of our speakers can answer some stuff. So Kalia, I think you are our next speaker. I think you're just on mute as well there.
Yes, okay, thanks. So right, you can all see my my screen now. Right, good. Okay, so hi everyone. My name is Kalliopi Panossopoulou. It's easier to just think of me as Kalia. So I'm gonna start giving a brief CV-like presentation of my work and my studies thus far. So I did a bachelor's in biochemistry and applied molecular biology at the University of Manchester Institute of Science and Technology. And then I did a PhD in yeast genetics. So I put there that it's a wet and dry lab because I was doing projects in the lab, which is the wet lab, but also quite a lot of data analysis, so computer-based work, which is what we tend to call dry lab sometimes for short. I then did my first postdoc, again working with yeast, molecular biology, so that was entirely wet lab project at the University of Manchester. I had a career break. I kind of not particularly liked the wet lab at that point. I've had a longish career break, and then I decided to return to a human genetics position. And I actually find it quite hard then to do this transition after a long break, but that kind of drove me to human genetic variation and statistical genetics of human common complex diseases, kind of similar to what Hilary as well has been doing. So I also studied at the Wellcome Trust Center for Human Genetics, Hilary. I was there for 6 months, probably before you, of course, much before you. And then the group that I worked with moved at the Wellcome Trust Sanger Institute. And then I had another career break for family reasons, and I decided to switch from a research role into a service role. So now I recently started working as a human trait curator at the European Bioinformatics Institute. So still in the campus, but in a slightly different role. Okay. So I think some of you may be interested to know what shaped my path and why did I choose to study what I studied. So I'm originally from Greece, and when I was about your age, or some of you anyway, I knew that I really liked biology and medicine, but I also liked maths, literature, and English. And the system in Greece was quite different, of course, than what it is in the UK. So towards at the end of our secondary education, the last year, you kind of have to select between 4 broad disciplines. So I went for medicine and biology, and there were 4 subjects that I had to take: biology, chemistry, physics, and essay, which is Greek writing. Unfortunately, no maths or stats. And probably you're gonna listen to me saying that several times throughout my talk, because I think there was a big mistake back then to not actually keep the maths going for longer. So for whatever reason, I failed to enter uni in Greece. I've had this idea with my parents to kind of go and study abroad. And I remember that my dad came one day and he said, "Oh, I heard that biochemistry is this up and coming specialty of biology, and perhaps maybe you can consider this." And I thought, yeah, why not? It's something more cutting edge, sounds good. And so I did a foundation course in biological sciences in Athens. The subjects were pretty much similar to what I already knew: biology, chemistry, physics, and English. Unfortunately, no maths or stats or even coding. So in my UCAS application, I remember I selected several universities. One that kind of I was quite keen to go to was the University of Manchester Institute of Science and Technology. I guess because it had a good rating. I liked the fact that the university was an institute of science and technology, and also because I liked Manchester music scene. So all these reasons brought me to study at Manchester, and I did my bachelor's, as I said, in biochemistry and applied molecular biology. I don't really remember the subjects that I did, but I remember that there was a lot of chemistry. There was quite a bit of biological subjects such as anatomy, immunology, and so on. And there were no maths or stats whatsoever. And there was, though, a good mixture of practical and theoretical and practical sessions, which I really enjoyed. During the 3rd year, for our 3rd year, actually, we had to choose optional lessons, and I chose the genomics and bioinformatics lectures that I really enjoyed. And that's when I talked to a professor who was doing some of these lectures about doing an MPhil in his lab. I was quite fortunate that the day after I spoke with him, he came back to me and he said, you can start working with me and you can also get a scholarship, which actually came from a home brewing company, because Professor Oliver was working was working with yeast, and he was actually the person behind the yeast genome sequencing effort that was published in '96 or so, and kind of carried on working with the yeast genome. So for my PhD, I was quite fortunate, as I said before, and perhaps envied PhD student, because I got to actually make beer and wine while doing my PhD, that not many people do. And so that's because, as I said, I was sponsored by this home brewing company who were interested in harvesting the potential of genetic engineering to improve the ethanol productivity of brewers' yeast strains. So I did quite a lot of lab work for them, but the other part of my project, which was the most interesting one and kind of set me up for what I ended up doing later on, was to analyze the global gene expression levels of yeast during a lager fermentation. So I would spend some time basically sampling and extracting RNA and doing this hybridization array experiments. And then I would end up with those primitive arrays, which were filters, whereby I would quantify the expression of all the genes in yeast using by measuring radioactivity. So then that meant that I was basically then moving completely into a computer and doing all my data analysis there and the interpretation into meaningful biological information. And I really enjoyed that part. Nevertheless, sorry, before I move into this slide, nevertheless, my first postdoc after my PhD was at a lab where I basically did pretty much more genetic engineering of yeast strains for a different reason, which I didn't particularly enjoy. And that's when I realized that actually I would like to go into large scale, more cutting edge data analysis projects in genetics. So, I mean, as Hilary pointed out before, the problem with common complex diseases is that you do have loads of variants that have a small effect size on increasing the risk of the disease. And so you need you need to analyze individuals with disease and compare their DNA with to individuals without disease, and that needs to be very large sample sizes in order to be able to find a link of a variant with disease. So I mostly work with many traits, but I mostly work with osteoarthritis, which at that time was thought to be more environmentally induced. And I mean, at that time it was probably 2008 when I started, and no one had found any links of genetic variants increasing the risk of osteoarthritis. So it was thought that it's a wear and tear disease, that as we grow older, our cartilage gets degraded because we move about a lot and because we carry a lot of weight, and basically that's what actually causes osteoarthritis, this joint space narrowing. But in order to actually, there were some genetic efforts to find variants for osteoarthritis, but the sample sizes were not large enough. So yes, when I joined the group where I started doing statistical genetics and looking at variants for osteoarthritis, that I was working for this consortium, which brought the expertise and the large sample sizes together in order to do the first, what was called genome-wide study for osteoarthritis, that was actually very fruitful and very rewarding. And I must admit that throughout the years that I worked as a statistical geneticist, one thing that I found extremely rewarding was it's a field that is very interdisciplinary, and it's a field that, as I said, you need loads of expertise and large sample sizes, so a lot of people get together to collaborate. So you have these passive collaborative international efforts that you're part of, which makes it very exciting and very rewarding in the end. So I thought of finishing with what my day-to-day schedule looked like, when I was working as in statistical genetics, I would spend quite a bit of time doing data analysis in Unix. I had to learn a little bit of programming to be able to do that. I would probably, I would get my results, whatever, and then I would pass them into several big biological databases to get to mine the data and proceed to biological information, or I would plot, plot graphs. Spend quite a bit of time writing publications, and you have to keep up to date as well while doing your work, because the science is so evolving rapidly, and so you have to kind of fit all of that, and also keep up to date by going to seminars, going to conferences, doing some courses, and brainstorming during meetings. Of course, I wasn't doing all of that in one day, but and that's what basically comprised most of my years that I was there. And no day was the same. Every day was different, and that is also very exciting. So I'm going to stop here, and I'm going to take any of your questions.
Awesome, thanks so much, Kalia. I'm just going to see if there was any specific questions for you. Someone's asked, how did you find the shift from yeast genetics to human genetics after your break from research?
That wasn't the toughest part, actually, because I didn't do that much yeast genetics in my undergraduate course. It wasn't that difficult. I mean, there are lots of differences between yeast and humans, of course, but genetics is genetics, and you can apply to different organisms. I think that what I found more of a steep learning curve, and that's why I stressed many times that there was no maths or stats, is that unlike Hillary, who did a little bit but wanted to do more, I did very little, and I stopped learning at the age of 17 before I even chose. I think at that time we kind of didn't know that genetics is going to be such a big data field. So that was more challenging than transitioning from organism to organism.
Awesome, thanks so much. I think we'll move on to our next speaker, but we'll take any more general questions for all the speakers at the end. So I think we have Carla next.
Cool. So I'm hoping that you can now see my slides. Great. So I'm Carla Jones. I'm a senior scientist at Sanger. And as the others in this panel, I'm going to talk a little bit about my career path so far and a little bit of the science that we do in our lab. So I am originally from Brazil. And as you can see, outside pandemic times, it's a very beautiful place to go, and recommend all a visit. And when I was in school, I had a very straightforward life plan. I would go to school, obviously, finish, get a biology degree, do a PhD in immunology, maybe go to a lab outside Brazil to do a postdoc, come back and land into a job in the local university. So I did finish school, I did get a biology degree and a PhD, And after my PhD, I decided to go to Imperial College in London. So at the time, I was interested to understand how cells in the bone marrow could contribute to the development of lung inflammation, especially asthma. And it was a really, really great time for me. I had access to technologies that you don't have access in Brazil, but it was also a very fun time. Scientists are very social creatures, as you can see by the pictures. And so much that I also met my husband at the time. That led to my 2 children. And because at the time, there was not much support for people to stay in academia, especially for people without family in the UK, I decided to take a career break. But I did know that my heart and what I really wanted to do was to go back to science. So in 2018, I heard about this returning to science fellowship. So many institutes noticed that there's a lot of scientists with care responsibilities that would leave in research just because of lack of support. And this kind of fellowship was bringing people back into science. So I decided to go back and I started a position in the Sanger Institute. So in this position, I am interested in look at complex disorders. And as you probably learn in school, monogenic disorders are, as the name would imply, caused by one gene leading to a disease. But as was mentioned before by Kylan and Hilary, in a complex disorder, you have a small effect size of several genes. And there are many diseases, many complex diseases, such as, for example, type 1 diabetes. It's not just type 1 diabetes. There are many complex disorders, especially associated with the immune system or with the dysregulation of the immune system, like IBD, which is an inflammation in the gut, or asthma in the lungs, or MS, for example. But how can we use genetics to understand why some people develop diseases and why some people don't? So to have a look at that, we need to go back a little bit to try to remind ourselves that the human genome is big. It has around 3.2 billion bases. And importantly, it has a lot of variation. So there are roughly 80 million different SNPs or different genetic variations in the human genome. So basically what we do in our department is trying to find this needle in a haystack. We are trying to find what are these small genetic variants that would be associated with increased risk for disease. So for you to have an idea, in early 2000, we had several studies, but there was just one region that was associated with increased risk for this disease called Crohn's disease, which is a disease associated with inflammation in the guts. But then the price of sequencing started dropping. So for you to have an idea, it started from hundreds and thousands of dollars, and now these days is as cheap as buying a couple of tickets to see Ed Sheeran. And it's not just money, it's also time. It used to take years and years to sequence a human genome, and now we can do that in a day. So because of that, we could increase the amount of people that we can sequence, and we can increase the power that we can, the power in the sense of the analytical power, that we can now detect many of these different regions that are associated with disease, which leads to another question. Right, we know that we have these genetic differences, but how do they really lead to disease? And this is the part of genomics that excites me the most, is the functional genomics. So we in the lab use a technique called CRISPR-Cas9 that was developed by two teams, one led by Jennifer Doda and another one led by Emmanuel Charpentier, that you can think as a really clever enzyme that can do two main things. One, it can identify a specific part of your DNA, and then it can cut it off. So you can, for example, delete a part, the area where these mutations or this genetic variation is on the genome. You can also delete disease candidate genes, and you can ask several questions. For example, on my little cartoon here, I am deleting receptor A. So I can then ask what happens to these cells without receptor A. Can we still activate these cells? Can these cells still migrate to where they should? And in our lab, we are specifically interested in a particular cell type called T cells that are central for the development of inflammation. So I'm bringing out this timeline, which I think is much more realistic than the timeline that I drew myself when I was in school. So it does look a bit more complicated, but involved much more. So I went to school, went to biology. I came to the UK to spend a year. This was 14 years ago. I fell in love, I had children. I went back to school. So during my career break, I went back to school as a secondary science teacher. I went back to science, and now I have a job as a senior scientist at Sanger. And that is the second thing that excites me the most about being a scientist, is being able to work with people from all parts of the world. So this is the team that I'm part of. And you can see that we have many people from many different countries. And I truly believe that all of this diversity really empower the science that we do. And I think that is my last slide, and I'm happy to take any questions. Thank you.
Awesome, thanks for that, Carla. If anyone's got any specific questions for Carla, do pop them in the chat box or the Q&A. I'll ask you a question before I see if any have come in. You drew a nice sort of straight, easy timeline back when you were sort of 17. If you could go back and tell your 17-year-old self one thing, what would you have told yourself at 17?
I would say just to, I think for you to reach to a good position, you need to have some planning. It's important for you to plan the steps, but it's very unlikely that all the steps will go as you planned, and that's fine. And that is much more fun, really, to have the surprises that life brings to you. And as long as you keep drawing new lines and new plans, it will still work out at the end.
Awesome. I think we'll move on to our last speaker, James, and then we'll take any more general questions at the end. So James, I'll hand over to you.
Okay, thanks. I will share my screen. Okay, so hopefully you can see that now. So yeah, I'm James Baker. I work at the European Bioinformatics Institute on the campus, and I'll go through sort of similar stuff to what you've already been talking about. So the key word is sort of bioinformatician. That's sort of the bit that I think we all do. So you've got your biology at the beginning, but we don't know what it's doing yet. Then there's people that go into the labs and test these things, and they come up with loads of data. And we sort of put that into something sensible that we can share and people can understand, that will hopefully lead to either more understanding or therapeutics or treatment plans, or it could be something that helps people. So I sort of went through similar broad stroke stuff to other people. I did my A-levels. I was always torn between English and biology, and what won out in the end was biology, because I was told that it's mostly reading and writing anyway. So yeah, that did it for me. I went through a process of clearing to get into my undergraduate degree. For maybe people not from the UK, that's where you don't get the grades that the university wanted you to get. So you have to go into this chaotic system over 24 hours where you have to sort of negotiate with other universities to take you on. Terrifying. Then I was an intern, did a master's, completed a PhD, and did a postdoc. That all looks nice and planned, but I think we've seen from Carla that it often goes a lot messier than that. So how much time do I have? I'll cut this a bit short. So just to echo Hillary's sentiment to do with sort of lab work. During my honours project, I was asked to crystallize a protein called Titin, which is the largest protein in humans. And we were just doing a small part of it. And crystallography is used because you get the crystal, shine some beamline through it from a particle accelerator, and the shadow it casts you can use to figure out what shape Titin was, which you can then use to get drugs and things to fit in properly. That's sort of the idea. Now, with crystallography, it's really hard to know how to crystallize a given protein. And I spent maybe 6 months, the best part of a year, and got no results whatsoever. It was horrible and I hated it. And it was monotonous, hard, long-houred work at funny hours. It wasn't for me. So I moved on to do, also associated with Liverpool, I went to Thailand and did an internship there, which was computational. And that was really a great experience. I think if anyone's ever got the opportunity to go and work abroad, I would thoroughly encourage you to do so. This is my supervisor, Dr. Tanyada Rungrat Monkol, and this is my assignment at a weekend. We went away and I was told to go and haggle for some fresh fish, which in Thai, and I don't speak Thai at all. So that was terrifying as well. I'll move on to, and then, so that sort of got me much more excited about science and going abroad in particular. So I applied for a PhD at Manchester as well. But also I spent 2 years of my PhD in Singapore, and this was my first real exposure. They had large compute power at the Thailand University, Chulalongkorn University, but this was a whole other level that Singapore had. It was if you could imagine something, you were limited in your imagination, not the compute power. Let's put it that way. And so I did lots of big data analysis, but still I'm nowhere really near genetics at all. I'm still in sort of the protein world. And that brings me to my current position at the European Bioinformatics Institute, where I work. This is sort of a much more collaborative placement that I've had than other institutes I've worked at. I'm working with clinicians and geneticists and mathematicians even. And we are essentially trying to look at these rare variants that are causing these tricky to diagnose diseases. And we're trying to interpret the molecular reason that causes the phenotype. So in a seizure patient that we had, if you imagine, we found a potential variant. And if you imagine sort of the neuron, and it's got to pass a signal along by maintaining a charge. And it uses pumps to move these charged particles from one side to the other very quickly so the signal can go along. This particular patient had a genetic variant that blocked these channels. So you can quite easily imagine how that would lead to sort of misfiring and dysfunction with the synapse firing. So yeah, it's a really interesting collaborative project that uses all sorts of different skill sets. Yeah, I think I've brought us onto time. So yeah, thanks for listening. And I think it'll be much more interesting to get people's questions than me bang on a bit more. So yeah, thank you.
Awesome, thanks, James. I'll start with a follow-up question that someone asked in the chat a while ago. I've seen that Hillary's replied in message, but I'll ask it to you as well, James. So someone asked what programs or languages you use to code.
So I personally use Python, which is really, really widely used and is becoming more widely used, but don't let that be. Most bioinformaticians I know use the language that they've are most comfortable with. It can be Perl, Java, anything. If you go into more service side of things, then they will have a specific language that they'll have in their job adverts to deal with their infrastructures.
Awesome. I don't know if anyone else wants to add to any other languages or coding programs that they use.
I think that for statistical genetics, then R is a very useful language to learn, which Hillary pointed out as well before. And I agree with that programming. I think Python is actually a language of choice now. Perl historically was used, and now more people are switching to Python, and it's actually more intuitive, I think, than Perl. But yes, it shouldn't be just a problem if you just learn another language.
Awesome. Yeah, I've popped some links in the chat for anyone who wants to look into coding. And I think from speaking to, I don't code myself, but having spoken to other people, I think just having a general understanding of coding will help with any language rather than being too worried about which one you're learning right now. Someone's asked you a specific question, James, and if anyone else wants to jump in as well. Someone's asked, did your PhD nicely lead into your current postdoc area, or did you sort of switch fields? And then more generally to everybody, like if you've switched fields a bit or moving from PhD to postdoc, how have you found that? So we'll start with James, but then if anyone else wants to chip in.
Yeah, so it was a bit of a pivot, going from sort of large, much more mathematical. In my PhD, it was much more mathematical, statistical stuff, and then going into looking at structures, very specific structures and things. And but fortunately, they're both focused on the same type of protein, so I didn't have to relearn all the biology that I was doing. So that was good for me. But I've heard of other people doing huge switches. I think it's a good point to switch. Yeah.
Anyone else want to chip in on how they found moving from PhD to postdoc?
I can add maybe to that. From my PhD to my postdoc was quite linear and quite straightforward, but once I had the career break, so my PhD and my postdoc were both in classical immunology. And when I came back and started as Sanger, I had to relearn all a different language about genetics and genomics and all different techniques. But learning is good. And, yeah, it's hard, but it did worth it, I think.
Awesome. There was another question in the Q&A that Hillary looks like you would like to answer, or anyone can chip in, but I'm aware that everyone we've got on our panel this evening has a PhD, so someone's asked whether a PhD is essential to go into this sort of line of science.
I mean, it depends what role you want. At our institute, there's a lot of people who work in a sort of service role, like generating large-scale DNA sequencing data or processing cell lines at large scale, or doing sort of software development to build software to handle genomic data. And so many of those people would not have a PhD. But I think if you want to be somebody who's actually asking the questions yourself and kind of intellectually engaged in trying to answer them, you generally, yeah, I mean, you need a PhD. But I think some people, before they kind of get to that point, think that PhD is kind of just another degree, and you sort of feel like at the end of undergrad you've had enough studying and swatting for exams and stuff, but it's really not like that. It's completely different. Doing a PhD is just doing research, but you get a bit of paper at the end that says you've done this degree. You know, it's just you're a junior researcher. That's my view. So, you know, if you want to do research, then you should do a PhD, I think, because there are some people who don't do that. You know, there's people who sometimes leave it till quite late before realizing, actually, if I want to progress in research, I need to do a PhD because I'm just hitting this block and I'm not going to be promoted. And PhD students are not paid very much. So I would recommend doing it early before you have kids and want to buy a house, because otherwise you may find, you know, you'll be shocked by the pay cut.
Excellent. Yeah, and I think there's also lots of different areas to work in science, and I think that historically PhD was the only access point to science. And whilst Hilary is absolutely right, if you want to progress it to very high levels of research science, a PhD might be the route you need to take. But for other areas of science, there are other ways of getting in. So I'm going to pop a link in the chat about apprenticeships in health and science. So if you want to work in a lab without being that sort of research lead, there are ways of doing that without going to university or without doing a PhD.
Yes, I think I would agree that I think I can see that there is a shift now towards apprenticeships and all that, whereby maybe people can in future do so without having a PhD. I think there is a shift from when I did my PhD, for sure, which was a long time ago, which pretty much everybody who wanted to pursue an academic career kind of needed to have a PhD, to that actually you don't necessarily need a PhD. There are other ways that you can get from A to C or D without a PhD.
Yeah, I think things are definitely sort of changing, and there are much more options for getting into all bits of science. On the sort of note of degrees, someone's asked whether anyone would advise studying a specific genetics degree at undergrad or doing a more general biology degree. I know that actually between the four of you have quite sort of a variety of different undergraduate degrees, but has anyone got any thoughts for that?
I mean, if you're pretty sure you want to do like that, you know, any genetics degree is going to have some molecular biology, some biochemistry, probably, hopefully some statistics. You know, you're not going to be doing pure genetics the entire time. It's just going to be genetics is a bit more the focus. So, you know, at one university, you could do as much genetics in a general biology degree as you would do in a genetics degree at another university. So I think what's more important is the content of the course and the choices you'll have, rather than what the degree is actually called.
Definitely. I think this has come up in sort of previous sessions that, yeah, having a look at what actually the course is going to offer you rather than the name is far more important. And if you're not 100% sure, then it's better to start with a more general biology degree. And if you like the genetics, just do more of those classes. But then actually, if you find that a different area of biology interests you more, you've still left that open. So yeah, have a look at the actual content of a degree, and then whatever you think you're more interested in right now, because it will be the next sort of 3 or 4 years of your life. So make sure you're interested in it before you start. that does bring us up to the end of our time for today. So thank you very much for all of our speakers for coming along and telling us about their careers. As I said, we've got one more session in this series that's going to look at the big questions, so some ethical and societal things to do with genetic variation. And you can sign up for that free. I'm going to pop that in the chat box now. You can also email us. My email address is in the chat box as well. If you've got more questions around anything that we talked about today, you can pop those through on an email, and I can reach out to our speakers to get some more answers for you. But thank you very much for our speakers, and thank you for everyone coming along, and hopefully we'll see some of you again next week.